So let me welcome our speaker for today, Ingvild Bjerke. Ingvild has a PhD in neuroscience and currently works as a postdoc in the Neural Systems Laboratory at the University of Oslo. There she combines her studies of the normal mouse brain development with neuroinformatics and digital atlasing. And according to herself, she is passionate about the open science movement and fair research and strives to make all data from her research public. And I believe that's exactly what we will be hearing more about today. So the floor is yours, Ingvild.
Thank you, Ingrid, for the introduction. I'll just share my screen. Like that. Yes, so already introduced, so I'll just start. In this talk, I will present the DOPAMAP project, a project aiming to provide a map of dopamine receptor positive cell types in the developing and adult mouse brain. So first, briefly on the background of the DOPAMAP project. This project was initiated during my PhD in 2019. And at that time, I was working on creating a database of quantitative cellular parameters in the murine basal ganglia. And I was searching the literature for relevant data. And in this work, I came across a publication by Ellen Collity and colleagues, which presented quantitative data of 2 dopamine receptor positive cell types from 5 brain regions in developing mice. And since I was creating a database of literature data, I was obviously reading a lot of papers every day at this time. And most of the time, I was just focused on extracting the information as efficiently as possible. But this paper made me stop for a while. And what was so striking about it was, first of all, the clarity and completeness of the documentation of the methods and presentation of the data. But most importantly, it was clear to me that the material behind this paper must be enormous, with immunohistochemical section data spanning 5 age groups, 2 cell types, and both male and female subjects. So while the authors had done a huge effort in quantifying a handful of brain regions, I was thinking about how much more there were to these data and how valuable it would be to have them available for the community. So I reached out to the corresponding author, Professor Ji Kim, to ask whether she would be interested in sharing the material. And she was, but could inform me that they did not have digital images of the material. And since we have a very efficient microscopy scanning facility here in Oslo, she agreed to ship all the sections by post. And we then scanned all the sections here in Oslo and started processing the material for sharing. And along the way, we also got funding support from the Human Brain Project through a voucher, and the collaboration has continued since then. So I'm telling all of this background because I think this project is a good example of how research data can be useful for so much more than the original hypothesis that they were produced to address. And I hope to convince you through the talk that data sharing can spark interesting and long-term collaborations that can give rise to so much more than a single publication. So without further ado, I'll go into the details about what we've done in the DOPAMAP project. So the aim of the DOPAMAP project was to integrate and share a large body of immunolabeled microscopy image data to provide an atlas of the developing mouse dopaminergic system. There are 3 main parts of the project. First, curation and sharing of all the data on Ebrains. Secondly, implementation of brain atlases for the developing mouse brain to facilitate interpretation and analysis of the data. And thirdly, semi-automatic quantitative analysis of all the material. And I'll go on to talk about each of these 3 project parts. So the first part of the project was to curate and share the datasets. And the data include brains from transgenic mice of the D1R and D2R EGFP genotype from both sexes and 5 age groups. So in total, 152 mice were processed using immunohistochemistry. And all of these data have been taken through pre-processing and curation pipelines to ensure that they are consistently formatted, named, and organized. Then they have all been registered to brain atlases to facilitate analysis. And lastly, both data and information about the data, which we call metadata, have been shared through Ebrains. So I'll briefly discuss each of these steps. So pre-processing of images includes renaming images so that they all have consistent names that make sense and that they are named after their anterior-posterior position in the brain. Then we do rotation in case some sections have been mounted with a slight tilt, like the one here, and flipping to ensure that sections are consistently oriented in terms of left and right hemispheres. And for these data, a small needle had been inserted in the left hemisphere, and you can see the hole here, so that it was quite easy to see whether a section had been flipped. And all of these steps can be performed with the Nutil tool, an open source software. After images had been preprocessed and organized, we performed spatial registration to brain reference atlases. And we used the QuickNet tool, which is bundled with the Allen Mouse Brain Common Coordinate Framework, a 3D atlas. And the atlas can be cut in any orientation to account for slight deviations from the coronal plane. So in this case, you can see that the section has been cut with a slight tilt across the hemisphere. And you can see that by the striatum here, for example, being larger on one side than the other. And as I said, we can adjust for that by cutting the atlas with a slight tilt across the hemispheres. Then generating a custom atlas map for each brain section. And we can also stretch the atlas to fit the dimensions of the section. And this is what we call linear registration, because the atlas is only adjusted in a linear way. And as you can see, there are some inaccuracies in the atlas, like for the cortex here and for the striatum here, where the borders do not exactly match the image underneath. And to adjust for this, we use a tool called Visualign, where different parts of the atlas map can be adjusted to exactly match the section. And this is what we call nonlinear registration. Both of these tools are also open access. And lastly, the images and their atlas maps were shared through the Ebrains knowledge graph. So if you go into the Ebrains knowledge graph and search for DOPAMAP, you can find this project shown here with 20 datasets included. And each dataset contains the data from one genotype, one age group, and either male or female subjects. So for example, here is a dataset for the adult, a dataset showing dopamine 1 receptor positive cells in the adult female mouse brain. So if you go into this dataset, you can see that you have metadata such as license, description, and keywords. You also have the option to download image files, or you can choose to view the files in an interactive image viewer. So if you click on one of these links, you are directed to a viewer showing you the images with atlas overlay that you can toggle on and off, and you can navigate the different sections in the series down here. And these images are high resolution and can be zoomed and panned interactively in the viewer. So an important part of the project was also to implement atlases for the developing mouse brain. The subjects in the youngest age groups were 17, 25, and 35 days old, and major brain regions in these subjects appear slightly different than in adults. So specialized atlases are needed to interpret them. And in the past, 3D atlases of the developing mouse brain have not been available, but more recently, Newmaster and colleagues created a set of developmental atlases by warping the Allen mouse brain common coordinate framework to serial 2-photon tomography templates of young mouse brains. And you can see an example of such a template and the atlas in 3D in this image. This is an example for the 21-day-old mouse brain. And to be able to register the DOPAMAP data to these atlases, we implemented them in XIII. So the last part of the project is to perform a semi-automatic quantitative analysis of the entire material. And here we use the Ebrains Quint workflow. And in this workflow, images are segmented using a machine learning-based tool called Elastic, so that each labeled cell is represented as an object, like these blue objects shown here. These segmentation images that result from this are then combined with the custom atlas maps from XIII, and each object is counted and sorted according to the brain regions they are located in using Nutil. So then we can create 3D visualizations like this one of each cell, color-coded according to the brain region in the Allen mouse brain atlas. And also, we can quantify the cells across the brain. Shown here are test data from the dopamine 1 receptor dataset from the adult male group. And each bar here corresponds to region in the Allen mouse brain atlas, color-coded as you can see here for the atlas. And on the y-axis are the number of cells for each of these regions. So as I said, this is only test data, and this analysis is still underway due to the large amounts of data in the project and the need for integrating the developmental atlases in all the tools of the Quint workflow. But we envision that the analysis will give interesting insights into the relative numbers of the 2 dopamine receptor neuron types across brain regions, developmental stages, and sex. So to conclude, through the DOPAMAP project, we have shared a large collection of high-resolution microscopic images showing dopamine 1 and 2 receptor positive cells in the developing mouse brain. We have implemented brain atlases for the developing mouse brain in XIII, and we plan to also implement them in the entire tool suite for the Quint workflow. We are currently working on quantitative analysis of the 2 cell types across the entire material. And lastly, I hope to have shown you that data sharing can provide opportunities for researchers to get much more out of their data than just a single journal article. So the DOPAMAP project has so far resulted in 20 datasets and multiple publications in preparation. And we hope that researchers all over the world will find a large collection of openly shared data useful in their own research. So lastly, I would like to acknowledge all the collaborators on this project. First of all, Professor G. Kim and Trygve Lergaard, Ellen Felty, who produced all the immunohistochemical material in this project, Casper and Crystal, who helped with data organization and sharing, and lastly, Hong, Gergely, and Maya, who has provided expert technical assistance on the project. And thank you all for your attention.
Wonderful, thank you, Ingvild. That was a very interesting talk, and I think a great example of open science in practice. I see there's one hand raised, but before that, I just wanted to ask, because you've taken in someone else's data, worked with this quite a lot, and then produced an output that's shared via the Ebrains platform. Could you say something about the challenges of taking over or presenting someone else's work like that?
Yes, well, I think one of the things that made it run quite smoothly in this project was what I said in the beginning, that Professor Kim actually shipped the entire material to Oslo so that we got to to scan them and digitize all the sections here, which made it very very easy to get an overview of the data. We got the list from them of all the metadata, and it has been, I think it's been a very nice story of things working very smoothly along the way. So what I will say is that Ellen Cullity, who prepared all the immunohistochemical data, is the first author on all the data sets, and I thought that was important for her to get the formal credit for all the data sets. And then in the upcoming publications, we will focus more on who performed the work presented in the actual papers. So I think it's worked nicely, mostly because everyone has been very open-minded. I don't think many people would be willing or would bother to send their data across the world physically. So I think it's been really nice.
It's for sure a very good example, for the whole research community, I think, to follow. An anonymous attendee asks, how much time did it take to prepare data for sharing as you did it?
Yes, so well, this has taken quite some time, and of course this was a huge data set. They were not intended for sharing in the first place. So we also have these quite elaborate pipelines with HBP registration and everything. So I think it took well, a few months at least to pre-process, organize, and share, or just to pre-process and organize the images, and then sharing is a separate process again. But I think that what's important to emphasize is that this process of sharing data will take considerably less time if it has been the plan from the beginning. So one of the reasons it took so much time is that we had to digitize all the sections from scratch. So if that is something that you do in the first place, then it's more a matter of staying organized throughout the process and taking care of all the information elements about the data. And then sharing will be quite easy. Yeah.
Great. I hope that was a sufficient reply. At least I think so. If not, please post another question in our Q&A chat. We have a next question from Abderrahman. As a senior medical student who is about to graduate, will I have any chance to work at a subject like this one? Maybe that's also touching on the time aspect, but also maybe also the type of data.
Yes, yeah. I'm not sure if I understand the question correctly, but if it is about the time aspect, I think again it's I think it's often quite a lot of work to organize and share data where it was not the plan to do that in the first place. And I have to admit, and I think that's something we all do, is that I definitely stay more organized when I know I will share something. So it's definitely something to think about from the beginning. Will I have any chance to work at a subject like this one? I'm not sure. Yeah.
So will I have to be enrolled in a PhD program to get access to this data?
Oh, no, you don't. The data are shared on the Ebrains knowledge graph, and everyone can download them. They are shared under a CC BY license, which means all you have to do is credit the data if you use them, but you can use them for whatever you want. Yeah.
Great, and he'll find these in the Ebrains, on the Ebrains platform. So you go to ebrains.eu, find data, and you can search by free text, for instance, and find Ingvild's data there. That's great. Okay, so you also showed us how you register these data to brain atlases. What would you say is the main benefit of doing so? What is the actual reason that you use these brain atlases in your data sharing effort?
Yeah, well, that's because it's a very good way to ensure that you can, or to be able to do more efficient analysis and to be able to compare data across subjects. And as I showed, the QuickNey tool can, in the QuickNey tool, you can cut the atlas in any orientation you want. So you can correct for these kinds of small deviations from the standard plane that we almost always get when we cut sections physically. It's virtually impossible to avoid that. And that means that it can be very hard to compare your actual sections to a traditional book form reference atlas. So that's the main advantage, I think, of the 3D atlases, that you, instead of trying to make your material look like the atlas, you make the atlas look like your material, and you get a higher kind of precision for that. And for this project, we also implemented the developmental atlases, which will fit the young brains better than an adult atlas, because they are slightly different when they are so young, and different brain areas develop at different rates, so they won't look exactly like an adult atlas. But then we can still compare across subject groups in a systematic way.
Exactly. Mm-hmm. So it is the first part of this workflow, or the analysis workflow that you showed us, would be this atlas registration. The next question asks if you can use the Quint workflow for rat data as well. So you showed
Yes. So I didn't show here since we only have mouse data in this project, but the Quint workflow is compatible with both rat and mouse brain atlases. The QuickNey tool, you can download different versions of it bundled with either the Allen mouse brain common coordinate framework or the Vaxall rat brain atlas, so that you can use either one of them for your analysis.
Are there atlases also for other species than rat and mouse?
Not currently implemented in this workflow. So then, but I'm sure there are other types of workflows for other species.
Good. One last question: Do you see any further use cases that these data can answer? Any further research questions that they can be used to answer?
Then what we are going to do ourselves?
Yeah.
I hope that these data will be very useful for more people than just us. That's the idea. I think they can be useful for anyone who's interested in the dopaminergic systems, either one of these two cell types, to be able to go to a high resolution microscopic viewer and look up different expression patterns for your regions of interest for these cell types. I think would be useful. Then there's also the option to download the data and do analysis. There might be people using other methods than we do for image segmentation. And if new atlases are made that someone would find more better in some way, then these data can of course be reanalyzed with different kinds of atlases. And I also think that the quantitative data that we are planning to extract from these data, they will of course also be shared, and I think those will be of high interest to researchers attempting to model the brain. So these are parameters that should be of interest to computational work. Yeah.
Great. So a bunch of potential for this data set that is available.
I think so, and I hope much more that I can't imagine today that someone will want to do in 10 or 20 or 50 years from now. That's the benefit of sharing, I think.
Cool. Okay, so a last question here before we round off. What would you say has been the greatest challenge for you when you shared your data? What would you recommend if I or the person who asked the question want to share my data as well with Ebrains? So one, greatest challenge, and two, what do you recommend if they want to share their data?
Yeah, so I think one challenge with this material was that, well, was probably mostly the pre-processing of all the images, and it's a small thing, but in the lab, when the sections were mounted on the glass, they were mounted very closely together, all of them. So that actually made it very difficult for the scanner to scan each section image separately. And this is the thing that did not affect the initial analysis for the authors who created it, but had a large effect on our work later. But so these are the kinds of things that one might not be able to anticipate, but I think the general remark or the general recommendation about it is to just stay as organized as possible during your work, no matter what you do, and do your work like someone is going to pick it up the next day and continue working on it. I think that's the main message.
That's a good reminder for many of us, I think. For other people, but also for our future self, I guess we could say.
Yeah, the future self as well. I must admit I have several times tried to pick up something I did a few months ago and not understood what I was thinking originally. So I'm still working on it myself to just always clean up when I'm done with something.
Good. Okay, so to round up, where can people find out more about Ebrains? Should they contact you?
About Ebrains more in general, I think.
Data, maybe. Yeah.
About sharing data?
Yeah.
Then I would go to ebrains.eu and navigate to the share data page, wouldn't you say?
Yeah. Sounds good. Perfect. Okay, so I think we'll round up there. There's some information in the chat, I see. The next Tea Slides is November 4th. Yeah, we're sharing screen. Perfect. So please apply. We are looking for more speakers. So yeah, hope to see you there. And the link to ebrains.eu is in the chat as well, which is perfect. Okay, thank you so much, Ingvild, for this great talk and good discussion after. Have a good rest day, everyone.
Bye.